regarding mitochondrial turnover with exercise in muscle. And the first speaker of this morning is Dr. Andy Philp from Centenary Institute in Australia. And he will present the title. Yeah. You have the word.
I will definitely present that. Thank you, Mark, and thank you to the organising committee for the invitation. This conference is really important to me, actually. This is my first ever scientific conference I attended as a first-year grad student in Maastricht a few years ago now, so that really kind of shaped my, I guess, changed the way I was approaching. I was an applied sports scientist at the time, didn't know anything molecular, had never seen anything molecular, and it really kind of blew my mind, that meeting, and I've been trying to catch up ever since. So it really is an honour to be here today to speak. So it would be remiss of me to start the session without really acknowledging that what we're going to talk about today is not new, right? So this is some work from 1915, and I think it's a fantastic example of, mitochondria have been on our mind as scientists for a long period of time now. So if you haven't read this, I would certainly encourage you to do this. This is some work from Lewis and Lewis. And these are drawings they were doing of mitochondria they were observing in cell culture at the time. And some text from this is really nice. So mitochondria, they identified really quickly are frequently arranged in the form of networks. And in those networks, they've observed all stages of formation in this living cell. And the important thing here is they were noting that the mitochondria were dynamic and they were changing, and their shape and their structure was continually evolving. And they also noticed this fact that there's these entire networks, they may break down into loops, threads, and granules, and you can see these here. So part of that dynamic change in the structure of mitochondria was the fact that there was a degradation or some kind of breakdown that was occurring. But at the time, they didn't really know what this was. So if you fast forward, and actually I should sort of talk about this, and the sort of summary of this, this paper, and I think there's still really, we don't know the answer to this now, which is also quite interesting, is that they asked a really open question, was what governs the arrangement of the mitochondria? Is it the shape of the cell? Is it the influence of the body or the nucleus? Maybe it's the internal structure of the cytoplasm, or even the metabolic activities of the mitochondria that changes the shape and the dynamics. So back in 1915, they already knew what was basically going on. So fast forward to the 60s, and this is some lovely work from Ashford and Porter, and they started to observe, and I guess the idea of this sort of degradation pathway was speculated, but this is an EM of some EM of the liver. And you can see quite nicely that they were starting to see mitochondria in lysosomal structures or structures that sort of had an autophagosomal formation. And this really was some of the first information, and you can see this here with engulfed mitochondria. There was some evidence of a mitochondrial specific degradation pathway or a mitochondrial autophagy that was occurring within tissue. And we know now that this is a selective specific mitochondrial autophagy program, or mitophagy. And this is, I think, a really nice schematic of describing what I'm going to talk about today. So we know the important thing here, there's a number of stresses that will actually change or isolate mitochondria, which will then, as this progresses, there's a recruitment of mitophagy receptors or ubiquitin autophagy receptors to the mitochondrial membrane. This is critical for this process because it allows the formation and the binding of the membrane to form a phagophore, which progresses to an autophagosome. There's addition of a lysosome, and ultimately you get a mitolysosome where you can degrade that mitochondria. So this is the process of mitophagy, and there's a huge amount of interest really in terms of the factors that drive this initial part, and what are the key sort of mechanistic machinery that will drive this process going forward. And importantly, mitophagy is linked to numerous diseases and numerous chronic diseases. And again, I think this is a lovely example of how you can look if there's an inactive mitophagy. Typically, you see an accumulation of damaged mitochondria, and then linked to this damaged mitochondria, neurodegeneration, tumorigenesis, tissue injury, inflammation, and aging. And there's some proteins that have been associated with these in mammals and flies and worms. The converse of this is stresses can also push this in a healthy way, so you can have active mitophagy. And that is also important because it can clean out the system and basically offset some of these events. So mitophagy seems to be one of those key kind of nodes where you can actually maintain a homeostasis of the mitochondrial pool and really offset some of this chronic disease progression. In the context of skeletal muscle, mitophagy really is a key functional component of what we now term as mitochondrial quality control. And I like this term because I think it encompasses the whole balance of the mitochondrial pool in skeletal muscle. So again, just as I showed you on that mitophagy diagram, typically there's some kind of stress response that's going to change the process of mitochondrial quality control in the tissue. That might change aspects of mitochondrial dynamics, where you're going to change the form and function of mitochondria to make the reticulum more efficient. And that's a process of fusion and fission that will sequester and remove parts of mitochondria, as well as elongate and promote mitochondria. Ultimately, mitochondrial protein balance is critical, and it's the seesaw of biogenesis and mitophagy to maintain the pool. And if you have all of these things in concert, you're going to have an increased mitochondrial function, and that's pretty much what we expect with exercise. So how do we measure mitophagy? And there's a number of ways that people have tried to do this. The most typical way people will do this in skeletal muscle is via mitochondrial fractionation. So you can take your human biopsy or your animal muscle, homogenize, spin down and fractionate the mitochondria, and then typically most people have looked at the recruitment of these autophagy or mitophagy receptors to that mitochondrial pool, and then assume that process would therefore indicate that mitophagy might be occurring. And this is an example. This is some lovely work from David Hood's group, not specifically to this study, but I think just an example of this process. So in this study, they were looking at exhaustive exercise, and then the black bars are 90 minutes in recovery from that bout. And you can just hear quite nicely there's an increased amount of LC2 proteins in the mitochondrial fraction with exercise. I think it's a nice example. This is not universal, so P62 wasn't accumulating to the same extent. But again, you can also see evidence of other sort of more mitophagy-specific factors, so in this case, the E3 ubiquitin ligase, Parkin, and a change in the mitochondrial ubiquitination. So I think as a tool and a technique, it's a very elegant way of looking at this in skeletal muscle. Of course, as things advance, Zen tends to do them. So this is some lovely work from Zen's lab. And this time it's using a fluorophore reporter. So this is Mitotimer, which is a lovely reporter. This is, I believe, an AAV where they transfected into muscles after exercise. And these reporters work really nicely, and I'll show you examples after this. But the idea is you have a green to red ratio. So as the proteins are degrading with oxidative stress, you can actually see a change in the green to red ratio that indicates a mitochondrial degradation. So you can see the change here of the red to green after exercise. And the red puncture is symbolic of mitolysosome formation as the mitochondria are degrading. The really nice thing that Rihanna Laker did here was actually co-localize the mitotimer with lysosomal protein. In this case, it's LAMP1, which is the yellow fluorescent protein. And you can see really nice co-localization. So the idea here is that as these mitochondria are degrading with stress, it appears it's happening in the lysosomal fraction, so therefore that is indicative of mitophagy. Now, it's always nice to see how these translate, and this is some work from Mark Franco's lab a few years ago now. Again, using the same idea of that fractionation, but this time in human skeletal muscle after exercise. This was the PhD of Cecil Schwang, and the interesting thing here is a really nice study design. It was fed versus fasted exercise. So you have a baseline post-exercise, or pre and post-exercise, and then 1 hour in recovery. And interesting here, in sort of contrast to the animal studies, there wasn't real evidence of a change in mitophagy in human skeletal muscle. So it seemed to be some differences. Ultimately, it's a semi-quantitative method, so there might be some differences there. But I think in terms of these techniques, it doesn't seem to translate quite as well into humans at this point. So that was acute exercise. There's some evidence that mitophagy might be involved in the chronic exercise training response. So again, this is work from David Hood's lab. So Carter et al. On the left-hand side looked at changes in, this is the intramyofibrillary LC3 flux. So this is the accumulation of LC3 in lysosomes. After, I believe it was 9 days of chronic contractile activity. So they showed in young, so this is 6 months, Fisher rats, there was a reduction in mitophagy after exercise, and that was consistent in the age group as well, although there was a hyperactivity of mitophagy in the basal state. So they also showed this this is in mice, this is wild type and Parkin knockout mice, again the same measurement after training. and mitophagy seemed to go down in the wild-type group and was pretty much abolished in the Parkin group. So I guess the summary from this work was, or the concept was that as mitochondrial biogenesis was increasing, mitophagy was going down, and that was why you had an increase in the pool size of mitochondria. So I'm going to basically present the work we've been sort of occupying ourselves for the last few years. So everything is unpublished, so I'm looking forward to a personal peer review. There's a few questions we wanted to clarify, and this is kind of I'm going to just give you a few snapshots of all of these today. So the first question was whether acute endurance exercise will increase mitophagy in skeletal muscle. You know, a simple start. And we've been using the MitoQC mouse. So this was described very nicely on day one of the meeting, if you were there, but I'll sort of remind you of what it is. So it's a mouse that was developed by Ian Ganizab, who's a collaborator in Dundee. Tom McWilliam was a lead author on this paper. It's a spectacular paper if you want to look at it. The reporter is a M-cherry GFP reporter that's a knock-in on the FIS1 protein, so it's on the outer mitochondrial membrane. And the way this works is it's very elegant that you have mitochondria that have this green and red tag, so the mitochondria are labeled in the basal state. And then upon a stimulus, like I showed you with that schematic, the process of mitophagy occurs. Once the mitochondria actually move to the lysosome and form the myelolysosome, the change in pH, or the pH shift here, you have a greater acidity of the myelolysosomes. That means that the GFP is quenched and you just retain red. So you can see the mitophagy occurring because you can see punctuate mitophagy or mitolysosomes. And you just get some beautiful autofluorescence in skeletal muscles. This is just a confocal image of, these are our muscle fibers. So you can see the lovely striation of the mitochondrial networks, and all these red puncture is the mitophagy occurring. You can do two things with that. You can then either look at the green to red ratio as a mitophagy index, or you can just measure the amount of mitolysosomes that are either in a total area or within a fiber, and that will tell you about the mitophagy occurring. So we've been working with these mice for a while, and you know, I'm always interested to see how far you can push a model and what you can actually do with it. So the typical way we'll look at these things is with immunohistochemistry. So I'll show you lots of images today from confocal work that we've done in fiber preps. But we also, at Garvin, where I was based in Sydney, there's a fantastic intravital imaging core. So if you're there, why not? So I'll show you some data around this, which I think is really quite exciting, so we can measure mitophagy occurring in muscle in real time. And I'll talk a little bit at the end about some of the flow cytometry you can do, because now that we've got these inherently labeled mitochondria, we can actually sort for these and then isolate fractions of mitophagy-specific mitochondria. So the first study I'm going to talk to you about is just an acute treadmill study. So we ran MitoQC mice. Everything we do is in male and female mice. But for today, in the interest of time, I'm just going to speak about the male group. So we ran them for an hour on a treadmill, incremental speeds. We've published that protocol previously. That was about an hour, and then we've got groups of 6 at pre and post, 30 minutes later, 3 hours later, and then 6 hours afterwards. So the idea was just whether or not we can see acute exercise changing in skeletal muscle. And the good news was that we could. So this is just a zoomed out version. This is a 400 times zoom in of here, and all I've done is just give you a grayscale of red just so you can see the punctuate mitochondria. So to talk you through this left to right, we have all of the graphs from here on are going to be control and then the time course. And at the bottom here, we have our mitophagy index, so that's your red to green ratio, and we can see a significant increase straight after exercise in these mice. And we're quite interested to see actually then it seems to go down and plateau, and then you have a later time point that comes up afterwards. So it's a biphasic change actually in mitophagy. If you calculate the total number of mitolysosomes, we saw a significant increase at that early time point. But then if you actually normalize that to fiber area, so you can make it more specific to the region, we saw a significant increase in the myelolysosome in the fiber area in both the immediately post and the 6-hour post. So an acute bout of exercise in our hands was able to increase mitophagy, and this is the gastrocnemius. So like I said, we were keen to see if we can look at that in a dynamic mode, and we're really sort of actively doing this, so you have to forgive me, we don't have a full data set. I was harassing my postdoc yesterday for more data, and he let me down. So So this is basically the model we have. So we run the mice for that 1 hour, anesthetize them, and then they're under isoflurane here, so they're on anesthetic. And you can do a couple of nice things there. So we're going to image directly on top of the muscle. So you can either basically sort of sock the leg and just do the whole muscle, or if you want to come back and repeatedly sample, you can just do a small incision on the skin, image the muscle, and then suture them up, and they can walk around thereafter. So on both of those occasions, we've just got the imager is basically just on the outside of the muscle. This is just a fixed image of what we see. So again, you see beautiful red and green. The SHG is really nice because it gives you the connective tissue and collagen. So what it actually helps us do is to work out the depth where we can get through the ECM and actually image the muscle. So this is the composite and the size that we'd look at. And then what we do is film basically for 30 minutes periods, and this is a composite again of the stacking that we do. So in that filming period, we can basically start and then work at depths of 5 microns down to 30 microns, and then we can cycle that as we go through. So hopefully this will give you an example of what we're doing. So on the top left here, when this plays, this is going to be the time lapse. This is going to be the depth, and this is basically the muscle that's imaging. I think we can play this if possible. So this is basically as we're scanning. So this is the time after exercise. So this is just in the basal state, so there's not much going on. So this is 10 minutes, and we're increasing depth. So we're constantly scanning through the muscle over time. So this is a 20-minute period. So all this acquisition is working really nicely to capture the green and red as it's going, and then it will loop. So that was our 30-minute time point. And then if we exercise them and then do the same thing, this is basically our muscle post-exercise. So hopefully you can still see on the screen that there's a lot more red puncture as we're actually scanning through the muscle. There's some nice organization of the muscle, and there's just a lot of really cool information going on. And Dean Campbell, who's the postdoc that's working on this, has obviously got a lot of data to work through, so he's been working up analysis techniques to do that. And what he's come up with, I think, is a really nice way of looking at this. So this is basically the Z-stack of the depth. So this is the periphery of the muscle down to the deepest that we go at 30 microns. This is the time across that 30-minute window. And then this is basically a basal change in the red to green, or the green to red ratio that indicates mitophagy. So the yellow here is indicating that as time increases after that exercise period, we're getting an increase of mitophagy that basically will increase in depth of the muscles we go through. So like I said, this is very preliminary data, but I think it's a nice tool that we can start looking at some of the dynamic changes of mitophagy in the context of exercise. So that's the acute work that we've been doing. But one of our real interests in the lab is actually how processes relate to chronic exercise training or chronic adaptation. So aim 2 really was to say, well, what's the relationship between mitophagy and adaptation to chronic exercise? So the model that we use is just a voluntary wheel running model. So mice get access to wheels for 21 days. We supplement all our mice with D2O so we can measure real-time fractional synthesis of mitochondrial proteins. I won't show you that data today. Like I said, they run voluntary for a period of time, and we have a group that we culled after 7 days and one after 21 days. I should note here that the 7 days, they basically run for 7 days, we take wheels away, and then we collect the muscle the following day. So all of this data is the chronic change. It's not affected by that acute or that last bout of running. And as you know, they run ridiculous distances. And the adaptation is fairly pronounced in this period of time, and we've published this model a few times. So when we look at this is the sedentary group. This is 7 days wheel running. This is 21 days. Again, this is your the zoomed out, zoomed in, and the green image. And what we saw again, we were very interested in is that as the mice adapt to training, you see a gradual increase at the 7 days, but then a significant change at 21 days. So this is the trained muscle. So this is the mitophagy in the basal state of that trained muscle. Okay, I hope that makes sense. So you see around about a 40% increase in mitophagy in the trained group, which again was interesting. And I won't show you the data today, but it parallels the change in synthesis and functional biogenesis in the muscles. So in our hands, I think the whole pool is increasing. So the overall mitochondrial turnover in the trained pool is greater. So this is all great. So we're seeing stuff going on. So the next goal we have and we're currently working on is to say, well, what might be regulating this change in mitophagy? As I highlighted earlier on, there's the the processes regulating mitophagy are fairly well characterized, certainly from the ubiquitin-dependent pathway. And it's quite exciting that there's a number of ubiquitin-independent regulators that are also being identified. So the classical ubiquitin-dependent model is that it works through a PINK1 Parkin pathway. So typically the idea is that under stress or damage, PINK1 is activated when a phosphorylate ubiquitin at serine 65, or it's good to remember because it's the same site as serine 65 in Parkin. This event will then trigger a number of things, but the key point here is that Parkin as an E3 ubiquitin ligase will actually bring ubiquitin to the receptors on the mitochondrial membrane, which work as scaffolds for a number of different autophagy receptors. So Michael Lazaro, when he was in Richard Yule's lab, showed quite nicely that 3 receptors are critical for this process, so optineurin, NDP52, and P62. And these allow LC3 proteins and GABRAT proteins to bind and form this autophagophore, which then initiates that process of mitophagy. In contrast to that, there's, like I said, there's some ubiquitin-independent pathways, and these are cool receptors that get recruited to the membrane and then work in the same way, so there'll still be scaffolds for LC3 GABRAT proteins, but they don't require ubiquitination to do that. And like I said, I think it's about 10 to 11 identified, but the key ones for people who looked at the skeletal muscle is FUNC1, BNIP3, and NIX. And again, I think the nice thing here as an exercise physiologist is there's a number of relevant stress pathways that we know change with exercise that might also be involved in this process. So we've knocked out basically all of these, but I'm just going to show you the data that we've got here today, which I'm going to start with is Parkin. So we've crossed our MitoQC mice with Parkin knockout mice. This is a whole body knockout. When we breed these as Parkin het MitoQC HOM, so we get, as you should, litamate controls to compare. And they're basically homozygote for MitoQC, but then we'll have wild type het or homozygotes for Parkin knockouts. So we ran these again for our 21 days with D2O, as I showed you previously. And we're quite excited. If you know any of my work, most of the time things just don't work. So it was quite interesting to see that for once we actually seem to have found something that is critical for exercise adaptation. So this is working through, we've now got a wild type sedentary, wild type VWR, het sedentary, het VWR, knockout sedentary, knockout VWR. And the really interesting thing is we were able to repeat the data I showed you previously, where the wild types will adapt to training, so you get an increase in, this is mitolysome total, mitolysome fiber area, but there was no change in both the hets and the homozygotes. So partial loss or full loss of Parkin completely blocks the ability of VWR to increase mitophagy. So we're actually kind of working out a lot around that, so I wish I had more, but we don't at this point. So I'll just finish with the studies we've been doing related to aging that have taken quite a while to do, so I'm happy to talk about. So the kind of key question we're interested in, and my group is very much interested in sarcopenia and age-associated changes in muscle function. And there's some really nice data. Actually, a grad student in the lab, James Slagar, wrote a nice review on this recently, talking about how changes in mitochondrial quality control might lead to this accelerated sarcopenic phenotype. I don't think it's certainly not the be-all and end-all, but it can certainly change the severity of this progression. And if you look in the literature, there's good evidence that there's potentially a suppression of mitochondrial biogenesis, and there's a number of pathways and regulators that might be driving this. There's less information in terms of dynamics, but there's some evidence, I would say less in humans. Very nice data in mice suggests that changes in the fusion-fission protein, or the ratio of those proteins, can really accelerate the progression of sarcopenia. Some fantastic work in that area. And there's some information around mitochondrial degradation, and again, changes in these markers or these drivers of mitophagy that might be changing. So we took our MitoQC mice, and we've got an aged colony of both male and female MitoQC mice. So I'm going to show you data from 3 month, 12 month, and 24 month. And then James, for his PhD, has basically trained the 24 months for 8 weeks. So this is slightly different. These aren't wheel run-ins. This is just treadmill run-ins. So they've done 3 times a week on a treadmill for 8 weeks at progressive intensities. So we've got pre and post to 6 weeks, and then we labeled them with D2O for the last 2 weeks up to 8 weeks. So it was a lot of work. He was in lockdown, so he was spending a lot of time with mice and treadmills. So the interesting data that we have from here is that mitophagy in our hands is going down with age. So this is 3 months, 12 months, 24 months, and then this is 24 months exercise. So if you look at total mitolysomes or mitolysomes per fiber area, now 3 months, and now 12 months, and now 24 months, we see a reduction in skeletal muscle mitophagy. And really exciting and interesting for us is that that 8 weeks of training almost restores the mitophagy to a young mouse level. So this isn't a lot of exercise, but it's a real remodeling of the skeletal muscle. So in our hands, mitophagy is sensitive to exercise, and it's able to change some of the process of aging. So to conclude the data that I've showed you, acute endurance exercise in our hands increases mitophagy at both early and late time points. The dynamic was quite interesting. 21 days of VWR will increase basal mitophagy in the trained state skeletal muscle. So as that muscle becomes trained, you get an increase in basal mitophagy. VWR mediated increase in the mitophagy are Parkin dependent. We're excited to explore this more. Aging results in a progressive reduction in skeletal muscle mitophagy, and we can rescue most of this with 8 weeks of exercise training, which is fantastic. And the final slide for me is just to say, how can we probe this more? And I think the really cool thing here is there's some beautiful data in using a FAMS model, so you can isolate mitochondria with facts. And this is just an example of this, where you can sort into quadrants based on the reporter. So the neat thing here is you can actually find an MCHA positive, GFP negative zone. So this would be our mitophagy zone here. And the protocol is really nice because you can fractionate that and then run proteomics on that pool, basically. So the idea here is that you can isolate the mitophagy proteome, and then we can start looking at the proteins that we recruited to that actively degrading that pool. So to finish, this is the people that have done all the work. So Ashley, Dean, and James at Centenary have really done a huge amount of the animal work. Like I said, at Garvan, we're very lucky to work with a number of different experts in intravital imaging, and Rob Brink is the mouse expert who makes our knockouts with a wonderful human being. At University of Melbourne, Paul Gregorowicz is helping us with some AAVs to tease apart some of the mechanisms. Mark Lawrence is helping us with total proteomics. Ian Ganley has been a long-term collaborator at University of Dundee, and Mike Lazaro and Grant Jewson at WeHire were kind enough to share the Parkin knockouts with us. I would love to take some questions, and while I am, I just want to highlight that there's a wonderful mitochondrial meeting that happens every 2 years in Australia called Aussie Mit. We're hosting this year in November, so if you've got money to spend on your grants and you want to come to sunny Sydney, we would love to have you, so please do come and see us. But thank you for your time.
Thank you, Andy, for this nice presentation. We have time for a few questions. Mark.
Lovely, Andy. Just with the last bit, with the aging, it really was quite remarkable data. What do you think the molecular mechanism is for that, for the exercise training restoration of the mitophagy? And I ask that question because I don't want to get into the murky pool of exercise mimetics, but a lot of older people, probably can't exercise. And if you could decipher a particular mechanism or pathway, it could be possibly druggable. Mm-hmm. So what do you think?
Absolutely. There's some lovely data, actually, and I don't want to steal the thunder of our later speakers, but if you overexpress Parkin in old skeletal muscle, you can actually rescue the sarcopenic phenotype. There's a very nice natural ingredient, Ubiquitin A, that's been, Johan Oryx's group published a lovely study showing you can nutritionally supplement old mice and old humans actually as well, and you get remodeling, and they link that to Parkin activation again. I don't know if it's robust in that model, but it seems if you can restore some of these mitophagy receptors, you can certainly activate mitophagy in old muscle.
One of the next speakers, when she was silly enough to work with me, published a paper looking at a link between heat shock proteins, and they actually decline with age, and they are somewhat druggable.
Yep.
Have you looked at those?
We haven't, actually. And I know that obviously BGP-15 was that kind of idea as well, right? So I think that the nice thing is there's lots of physiological stresses, certainly, and heat shock proteins, I think, are certainly those that exercise sensitive and can definitely model. There's a whole HIF1 pathway as well, I think it's quite interesting. So there's a lot of cool things.
Cheers, thanks.
Thanks, Mark.
Thank you.
Hi, Yun Tian from University of Virginia. So I really liked your experiments. I think they're very, very exciting. I just have one quick question about the mitophagy after training? Because before all those research, we know that training will induce, make the mitochondria better and maybe more from biogenesis. So I feel like if mitophagy is a stress response, wouldn't muscle need less mitophagy, right, after period of training? So you had the 21 days and I think also the 8 weeks training showing that there's increase in basal mitophagy. I just wonder whether you have any interpretation around that kind of maybe it could be a paradox or maybe it's just, I don't know, philosophical question more.
No, great question. I think, so the way I look at it, I think, sometimes we can think of autophagy or mitophagy is a bad thing, right? It's there to sort of, if it's high, going to degrade and it's a negative. I think the other way around is that it's turnover, and the whole point is trying to keep the system active and healthy. So in that model, we see about a 30 to 40% increase in biogenesis. We see about the same in mitophagy. So in a trained muscle, our interpretation is the whole system is better. The whole thing is working more. So your basal state is higher, so therefore your turnover is higher and your functional pool is higher. So that's the way I sort of reconcile that data, that you don't need to have an imbalance because the whole thing is moving together and the whole system is therefore healthier.
Okay, thank you.
Thank you. Last question.
Hi, Gilles Gospiou. Thank you so much for your talk. Just a quick curiosity question. Have you looked if there is fiber type specificity in the response? And because you use male and female, is sex possible dimorphism, sexual dimorphism?
Yeah, we haven't done the fibers. All of this work was in the gastrocnemius, which is, as in mice, right, it's typically fairly fast muscle anyway. There's a fair bit going on with male to female that I don't have time to talk about. But there's that was why we wanted to do it, just to see if there was a difference. And there seems to be a few, not dramatic differences, but subtle differences that I think are quite exciting. So hopefully I can share that another time.
Thank you so much.
Thank you. Thank you, Andy.
My pleasure.